Restriction endonuclease. Now this restriction endonucleases are the bacterial enzymes that cuts the DNA into fragments, and they cut the DNA after recognizing a specific nucleotide sequences known as recognition site or restriction site. So restriction endonucleases recognizes the restriction site, and on the basis of that, then it cuts the DNA into fragments. That is how the restriction endonuclease works. Now, in the term restriction endonuclease, the term restriction represents that these enzymes known as restriction enzymes restrict the entry of foreign DNA into bacterial cell. And that is the reason why they are known as restriction enzyme, because they are restricting the entry of foreign DNA into the bacterial cell. Okay, it is believed that these restriction enzymes have evolved in bacteria to resist the viral attack. Now, the existence of restriction enzymes were first postulated by W. Arber. Okay, this scientist first postulated the existence of restriction enzyme. He observed that when DNA of bacteriophage enters the host bacterium, then that DNA of bacteriophage is cut up into smaller pieces. And on the basis of this observation, he theorized the presence of restriction enzymes. So bacteriophage is infecting host bacterium, then that bacteriophage is inserting its DNA into host bacterium, but that host bacterium is cutting the DNA of bacteriophage. And this observation was made by this scientist, and then he theorized the presence of restriction enzyme. Hamilton Smith and his co-workers were the first one to isolate the restriction enzyme from the bacterium known as Haemophilus influenza strain RD. So these scientists isolated the restriction enzyme for the first time, but its presence was theorized by W. Arber first. Now, enzyme known as Hind2 recognizes 6 base pair as of the DNA sequence. A 6 base pair DNA sequence is recognized by this restriction enzyme known as Hind2, Hind2. And this was the first restriction enzyme that was isolated. So after the discovery of this restriction enzyme, EcoR1 was the restriction enzyme that was isolated and characterized from Escherichia coli strain RY13. Now let's talk about the nomenclature of the restriction enzymes. Now the name of restriction enzymes is consisting of 3 parts: an abbreviation, letter, and a Roman numeral. Now this abbreviation is of genus and species of an organism from which the enzyme is isolated, and that abbreviation is of 3 letters. For example, ECO for Escherichia coli. E represents the genus and CO represents the coli, CO coli. So this first letter in this abbreviation is representing genus, and these next 2 letters are representing species. So that is how the abbreviation part of the name works. Now after that, after abbreviation, the name of the restriction enzyme contains a letter or a number or a combination of letter and number both, which indicate the strain of the relevant species. And after that, you have Roman numeral, which represents the order in which the different restriction modification systems were found in same organism or in same strain. So in the name of the restriction enzyme, you have an abbreviation, then you have a letter, number, or combination of both, and at last you have a Roman numeral. So for example, EcoR1, E represents Escherichia, which represents the genus, of this organism from which this enzyme is isolated. And then CO represents coli, which is the species of this organism. Then R represents RY13, which is the strain of this organism. And 1 represents first identified, that is the order of identification of the bacterium. So that is how this restriction enzyme EcoR1 is named. Now let's discuss about restriction sites. Now a restriction enzyme don't go and cut randomly. Okay, it cuts only at the double helical segments of a particular nucleotide sequence of 4 to 8 base pairs, which are known as recognition sites or restriction sites. Okay, the restriction enzyme will only cut on recognition site or restriction site, which is a particular nucleotide sequence of 4 to 8 base pairs in the double helical segment. Now this recognition sites or restriction sites are generally palindromic sequences. Okay, so this restriction enzyme cuts only at the palindromic sequences. Now the position at which the restriction enzyme cuts a particular sequence of DNA is symbolized as this slash. It will be explained in further example how exactly this symbol works. Now when restriction enzyme cuts a particular DNA, it makes either blunt cuts or staggered cuts. Okay, it makes two types of cuts: blunt cuts and staggered cuts. Okay, so restriction fragment that is generated by the action of restriction enzyme may have a blunt end or it may have overhanging end. Blunt end means the cleavage point is exactly at the axis of symmetry, and the overhanging ends means the cleavage points don't fall on the symmetry axis, and the restriction fragment that is generated have sticky ends or cohesive ends. Okay, these overhanging ends are also known as sticky ends or cohesive ends. And what does it mean? It means that the cleavage, the point at which the cleavage is taking place, don't fall at the symmetry axis, and in case of blunt ends, the cleavage point falls exactly at the axis of symmetry. So this is how the blunt end and the staggered cut or the sticky end looks like. Okay, you see, this is the cleavage site. This is where the restriction enzyme is cutting the DNA fragment, and this is that restriction enzyme ECOR5, and when it cuts at this cleavage point, then this is how the particular fragment is splitted up. Okay, it has blunt ends. Okay, exactly the cleavage point is at the axis of symmetry. But in case of this staggered cuts, the cleavage point don't fall at the axis of symmetry. This is how the staggered cut look like, and this is how the blunt end look like. Okay, so yeah, when EcoR1 cuts a particular DNA fragment, then this is how it is cutting. These are the two different cleavage sites, and they are not perpendicular. They are not at the same axis. But in this case, both the cleavage sites are on the same axis. But in this case, they are not on the same axis, and that is the reason why they create a staggered cut. And when the cleavage sites are on the same axis, they create a blunt cut. Now, after a staggered cut, when the cleavage sites are not on the same axis, the resulting restriction fragment may have a 5' overhang or a 3' overhang. For example, consider this restriction enzyme known as EcoR1. its restriction site is GAATTC. Now when this restriction enzyme works and the staggered cuts have been made, the resulting fragment have 5' overhang or 5' staggered ends. Okay, we will see how it looks. And on the other hand, when restriction enzyme known as PST1 works on a particular DNA molecule, then it generates staggered cuts which are known as 3' overhangs or 3' staggered ends. So this is what we are trying to say. Okay, EcoR1 worked on this particular sequence, and when it made cut, it produced two staggered ends, and as you can see, this is a 5' overhang and this is also a 5' overhang. But when this enzyme PST1 made a cut on this particular sequence of DNA, then it generated a 3' overhang in both the case. So yeah, in case of the staggered cuts, staggered ends, the ends can be 5' overhang or it can be 3' overhang. It is not going to be like one is 5' and another is 3'. No. Either it is 5' overhang or it is 3' overhang. That is how it is going to work. And it will depend on the restriction enzyme and how it is making cleavage at the restriction sites. So remember this, that EcoR1 is producing 5' staggered ends and PST1 is producing 3' staggered ends. Now in non-standard conditions such as low ionic strength or high pH, there are some restriction enzymes which cleave a particular sequence which is similar but not identical to the defined recognition sequence. Okay, each and every particular restriction enzyme cuts on the defined recognition sequence, correct? But in some non-standard condition, that particular restriction enzyme can cleave a sequence which is not identical to this defined recognition sequence, but it is similar. And this altered specificity of this restriction enzyme is known as star activity. Okay, so what is star activity of a particular restriction enzyme? Star activity is the ability of a particular restriction enzyme to cleave a sequence which is similar but not identical to the defined recognition sequence. That is what the star activity is. Now this star activity is a general property of any particular restriction endonuclease. Any restriction endonuclease will cleave at non-canonical sites in certain extreme conditions, and that is what the star activity is all about. In non-standard condition, you cleave a particular sequence which is not identical to that particular recognition site for which that enzyme works. Now let's talk about isochizomers, neochizomers and isocodamers. Now these isochizomers are the pairs of restriction enzymes that recognize the same recognition sites and cut at the same location. Okay, for example, this SPH1 and BBU1, these two are the restriction enzymes which are recognizing this same sequence and are cutting at the same location. Okay, this slash is representing that the cut will be made between C and G. Okay, so that is how you represent the cleavage, by slash. And that is the reason why this SPH1 and BBU1 are known as isochizomers of each other. They are recognizing a same sequence and they are cutting at the same location, isochizomers. Then you have neochizomers which recognize the same sequence but cut differently. For example, this SMA1 and XMA1, both of them are recognizing GGG-CCC, GGG-CCC, but they are cutting at different site. Okay, SMA1 is cutting between G and C, but this XMA1 is cutting between G and G. So that is the reason why these two enzymes are known as neochizomers of each other. They are recognizing the same sequence, but they are cutting differently. And then you have isocodamers, which have slightly different recognition sequences, but they produce same ends. Okay, these isocodamers are the restriction enzyme, different restriction enzymes that have slightly different recognition sequences but produces the same ends. For example, this SAU3A, this restriction enzyme, and BAMH1, this restriction enzyme, both of them produce GATC sticky ends, but they have a different recognition sequence. Okay, the recognition sequence for SAU3A contains 4 base pairs and the recognition sequence of the BAMH1 consists of 6 base pairs. Okay, so they have slightly different recognition site, but after their activity, after they cleave the particular DNA, same sticky ends are produced, that is GATC. Okay, so this is how it goes. Okay, so this is the recognition site for BAMH1. Okay, this whole, and after it cuts, it produces CTAG, this sticky end, and for SAU3A, this is the recognition site which is smaller than this one, but after this SAU3A works, it cleaves, it also produces CTAG, hanging, staggered end. So both of this enzyme are producing same kind of staggered end, even though they have slightly different recognition site, and that is the reason why they are known as isocodamers of each other. Now let's talk about the frequency of recognition sites. Now the number of recognition sites of a particular restriction endonuclease in a DNA molecule of known length can be calculated mathematically. Okay, there is a particular restriction endonuclease, there is a particular DNA molecule of known length. Now, how many recognition sites are there in this DNA molecule of known length for this particular restriction enzyme? That number of recognition sites can be calculated mathematically. Now assume that the nucleotides are ordered in a particular DNA molecule in a random fashion, and the 4 different nucleotides are present in equal proportions also. Then the recognition site of 6 base pair will occur at a frequency of 1/4:6, which is 1:4096 bases. It means that if this is your particular DNA molecule and this fragment is made up of this 4000 base pair, then at each 4000 base pair you will have one recognition site which is made up of 6 base pair. Okay, that is what it means. So ECO R1 recognizes a recognition site of 6 base pair, that is GAATTC. It means when ECO R1 will cut a particular DNA, it will produce fragments of 4 kb. Okay, this is an estimate. For example, consider this only. Okay, so this is your DNA molecule, okay, of a particular length, and it is subjected to echo R1, and echo R1 recognizes the recognition site of 6 base pair, and the 6 base pair is going to come after each 4000 base pair. So this is the recognition site, then this is the recognition site, then this is the recognition site, and this is the recognition site, and each of them have a length of about 4000 base pair, that is 4 kb. Okay, so when this restriction enzyme will work, this DNA molecule will be converted into DNA fragments, and each DNA fragment will be having a length of 4 kb. Okay, each DNA fragment will have a length of 4 kb when it is digested with echo R1. And why? Because the frequency at which its recognition site will occur on a particular DNA is estimated about after each 4000 base pair. Okay, in each 4000 base pair there will be one recognition site for echo R1, because it is made up of the recognition site of echo R1 have 6 base pairs in it. Now this is an estimate. Okay, this 4 kb fragment will be produced by the action of echo R1. This is just an estimate. The actual size of the restriction fragment that is produced by the digestion of a particular DNA molecule with a particular restriction enzyme vary considerably. Okay, it is not that always 4 kb fragment will be produced. In practice, it varies a lot. It is just an estimate. Now, for example, hint 3. Okay, this restriction enzyme recognizes a 6 base pair sequence known as AAGCTT, and on the basis of this information we can calculate its frequency 1 in 4096. So if this is a particular DNA fragment, then after each 4 kilo base pairs it will make a cut. After each 4 kilo base pair it will make a cut. That is what we are estimating from this information. Okay, but in practice, when bacteriophage lambda DNA, which is made up of 48502 base pair, that is about 48,000 base pair, when this DNA of bacteriolambda is subjected to hint 3, then it produces fragments which vary in length from 123 base pair to 23,000 base pair. Okay, on the basis of this estimation, only 4 kb base pair should have been generated, but in actual practice the fragments varied in length from 123 base pair to 23,000 base pair. So that is how it works in practice. Okay, so that's all for this discussion. This thing is quite hard to digest, but let me explain you what I understood. So this is your particular DNA fragment. For example, consider it is made up of 40,000 base pairs. Okay, now the recognition site that a particular restriction enzyme will recognize is made up of 6 base pair. Okay, now by calculation we estimated that after each 4000 bases, okay, this particular 6 base pair segment will occur. Okay, after each 4000 bases, and that is the reason why if you cut this DNA with that particular restriction enzyme whose recognition site is made up of 6 base pair, then it will theoretically should produce 10 fragments which has length of 4000 base each. Okay, so 4000 into 10, you will get 40,000. This is theory, but in actual it does not happen that way. That is what this discussion is all about. But you get a estimation, so that is the reason why this formula is also important. Thank you.